Thank you. Uh, my name is José Onuchic. It's an honor for me to join this academy, and I thank you for the confidence you placed on me by this choice. It's a pleasure to see in this group of new members my good friend Francis Arnold. I was born in the city of São Paulo, Brazil, but I grew up in São Carlos, where my parents were both professors of mathematics in the local campus of the University of São Paulo. In addition to be a family very involved in academia, we were devout Catholics. My father went to a Catholic seminar for 10 years. Sorry. I'm sorry, just to turn this off. My father went to a Catholic seminar for 10 years earlier in life. At the University of São Paulo in São Carlos, I obtained bachelor's degrees in electrical engineering and physics, followed by a master in science and applied physics in 1982. I studied for my PhD at the California Institute of Technology, Caltech, with John Hopfield. After graduating from Caltech in 1987, I returned to Brazil as an assistant professor at the Institute of Physics in São Carlos, University of São Paulo. At that time, my wife and I decided we were going back to Brazil for good. This just lasted a few years. In 1999, I came back to the USA, where I was recruited by the physics department at the University of California, San Diego, to establish a theoretical biological physics group. Since 2002, I have co-directed the Center for Theoretical Biological Physics. CTBP is an NSF Physics Frontier Center that encompasses a broad spectrum of research and training activities at the forefront of biology and physics interface, including molecular biophysics, statistical biophysics, genome structure and function, and biological dynamics at the cellular and multicellular scale. The interdisciplinary approach includes physicists, chemists, mathematicians, biologists, and has created a better understanding of the underlying mechanisms governing complex biological systems. In July 2011, my group moved to Rice University, where I relocated my Center for Theoretical Biological Physics. Currently, I'm the Vice Professor of Physics and Astronomy, Professor of Chemistry, and Professor of Biosciences. I also the co-director of CTBP and a Cancer Prevention Research Institute of Texas Scholar in Cancer Research. My research looks at theoretical and computational methods for molecular biophysics, chemical reactions in condensed matter, genome structure and gene expression, gene networks, and theoretical modeling of cancer. My first contribution focused on the theory of chemical reactions in biological electron transfer. These reactions are central to the bioenergetic pathways in living organisms. In the mid-1980s, I created the concept of tunneling pathways and the methodology which describes proteins as a combination of relevant tubes of pathways that provide new ways of designing electron transfer proteins. My theoretical framework has been confirmed and utilized by many experimental groups. This combined theoretical and experimental effort has substantially improved the understanding of electron transfer process in biology and also chemistry. In the 1990s, we created a new view for protein folding. My research group introduced the concept of protein folding funnels to explain how select sequence of amino acids, proteins, can fold into a unique three-dimensional structure. Convergent kinetic pathways, or folding funnels, guide folding to a unique, stable, native conformation. Energy landscape theory and the funnel concept provide the theoretical framework needed to pose and to address the questions of protein folding and functional mechanisms. These theoretical advances have guided much of the experimental research in protein folding during the last 3 decades. The impact of energy landscape theory and the funnel concept is not limited to protein folding. Cells contain a large number of molecular machines that control and maintain cellular functions with exquisite precision. We have used this energy landscape theoretical approach to understand the underlying mechanisms governing molecular models, such as kinesin and myosin. During the last 15 years, we expanded our scientific interest towards modeling genetic networks with emphasis in bacteria and cancer. In nature, bacteria live in large colonies. We have been interested in understanding how bacteria make decisions using a specialized network of genes and proteins. This network enables bacteria to make choices when faced with different stresses and environment conditions. Generalizing and expand these ideas about decision-making to mammalian cells has moved my group towards searching for mechanisms governing cancer. It's important to highlight that mechanisms and networks in mammalian cells are much more complex and sophisticated. After joining rice, we further extend our landscape ideas towards folding and function of the genome. We are currently very interested in understanding how the genome structure and dynamics affect gene expression and other functions in the cell's nucleus. We have a great experimental partner, Aries Lieberman Iden, that works with Eric, that we're talking about, an entire new field, an entire new life start with this new field, and I hope to connect it with our work in cancer modeling in the near future. Thank you very much. I'm looking forward to engage in academic activities and help with its mission.